and welcome to Miss Estruch Biology and this entire Topic 6 theory video. And if you want to make notes as you go in a structured way, check out my free workbook in the description that you can download now. If you want even more help though, for example, me being your teacher, plus 2 other highly experienced examiners and teachers teaching you as well, then don't forget to check out the Biology Study Club. This is my monthly membership where I give you tuition online, some of my resources for free, you get exclusive discounts, you get mock papers marked, you get other work marked, plus so many other benefits. Then check out the link in the description to join the Biology Study Club and maybe I'll see you there. AQA Topic 6, and we begin with 3.6.1 Stimuli, and this is looking at the internal and external that can be detected and lead to a response. Starting with your definition of a stimulus. This is a detectable change in the environment, and these stimuli are detected by cells known as receptors. And an organism increases their chance of survival if they can respond to stimuli via different response mechanisms. So for plants, there is response as well. And we begin by looking at tropisms, which is the term given to when a plant responds to a stimulus and they respond via growth to that stimulus. A positive tropism means that the plant would be leaning towards or growing towards the stimuli. Negative means moving or growing away from it. And the 2 that you learn about are the stimulus of light and gravity. Tropisms are controlled by specific growth factors, and the one example you learn about is indole acetic acid, or IAA, and you can abbreviate to that in the exam. An IAA is a type of auxin, and therefore it can control cell elongation in the shoots and inhibit the growth of cells in the roots. It's made in the tip of the root in the shoot, but then it diffuses to other cells within the shoot and the root. So if we have a look at phototropisms first of all, photo meaning light, so this is the response to light. In the shoots, light is needed for the light-dependent reactions that you learn about in topic 5. So the plant is going to benefit by gaining more light energy and therefore leaning or bending towards the light source, the stimulus. And this is positive phototropism. So the shoot tip is where IAA is produced. IAA will diffuse to the shaded side. So if you've got light from one side over here, the IAA will diffuse to the shaded side. And that is what we call unilateral light source, and therefore we get this uneven distribution of IAA. And the cells that are exposed to the IAA are going to elongate more, and as a result the shaded side cells will be longer than the light side, so it's longer and it bends over towards the light source. In the roots don't photosynthesize, they don't have chloroplasts and chlorophyll, so they don't require light energy. So there's no advantage of growing towards the light source. And instead, there's actually an advantage to grow away, because that means the roots are anchoring the plant deeper into the soil. And in the roots, if cells are exposed to a high concentration of IAA, that inhibits cell elongation. So you still have IAA being produced in the tips of the root, but the IAA diffuses to the lower side, so the direction where you would have gravity, the lower side, and that inhibits the elongation of the cells on the lower side of the root. The ones on the top side are still going to grow, and therefore that top side is longer and it bends downwards away from the light source and towards gravity. But this would be our negative phototropism because it's away from the light source. With regards to gravitropism, in the shoots, again, IAA is produced in the tip of the shoot, and IAA will diffuse from the upper side to the lower side of a shoot. And if a plant is vertical, not lying down like we can see in this picture, this causes the plant cells to elongate and the plant grows upwards because the IAA would be moving down towards the bottom and those bottom cells will grow more, and it just makes the shoot grow upwards. If a plant is on one side though, like we can see in this picture, the side that is exposed to the auxin will elongate and that can cause it to grow upwards. And that would be negative gravitropism, it's growing away from the direction of gravity. In the roots, it's the opposite. The IAA will diffuse to the lower side of the root so that the upper side elongates and the lower side doesn't, so the root bends downwards towards gravity, which is a positive gravitropism. Next then, the other simple responses are taxis and kinesis, which keep organisms within their favourable conditions of their environment, for example, light intensity, moisture levels and chemicals. Taxis, this is when an organism will move its entire body towards a favourable stimulus or away from an unfavourable stimulus. So this is directional. When an organism moves towards a stimulus, this is known as positive taxis, and when it moves away, it's negative taxis. Kinesis then, this one is not directional, but it's a change in the speed of movement and the rate it changes direction. If an organism moves from an area where there are beneficial stimuli to an area where there are more harmful stimuli, its kinesis response will be to increase the rate it changes direction, and therefore it's more likely to locate and return to the favourable conditions again. However, if it tries that and the organism is completely surrounded by negative stimuli, so no matter how many times it changes direction, it can't find favourable conditions, then the rate of turning actually decreases to keep it moving in a relatively straight line to increase the chances of it finding a new location with favourable conditions. And just linked to this, most of the responses will be that they'll want an area where it's dark, and that would be to not be easily seen by predators or to stop them from drying out. Moisture, they'll want a more moist area, again, to stop them from drying out. Chemicals, they might be moving towards certain chemicals because that might indicate the presence of food. Simple reflexes are something that we have in more complex organisms with a nervous system, and this would involve a receptor detecting a stimulus. That would then result in coordination occurring with the coordinator, then a response carried out by an effector. And the nervous system is made up of the peripheral and the central nervous system. The peripheral, abbreviated here to P, nervous system, NS, is the receptors, sensory and motor neurons. The CNS, or the central nervous system, that's the coordination centres, so the brain and the spinal cord. So here we have a simple reflex, and a reflex is a rapid automatic response to a stimulus. Reflexes are involuntary, so they do not require conscious thought. You don't have to learn to respond in that way to protect yourself. And the point of a reflex is it protects against damage to body tissues, and it could also help you to escape from a predator. There are 3 neurons in a reflex arc, so it's very, very simple. You have the sensory neuron, which is going to be connected to the receptor. That detects a stimulus and sends an impulse to the spinal cord. The relay neuron is going to be located in the central nervous system, which will be the spinal cord for a reflex. And then the motor neuron that carries the impulse to an effector to carry out the response. And the response in terms of a reflex reaction is very fast because the impulse bypasses the conscious part of the brain and it travels through the reflex arc. And as we can see, the reflex arc only contains 3 neurons and therefore 2 gaps, 2 synapses. And because we've only got 2 synapses, that means it's faster because it's slower transmission at synapse because it's diffusion of a neurotransmitter. Now, actually, in this image, you can't see the relay neuron, but there would be a sensory neuron, relay neuron here in the spinal cord, and then the motor neuron. Receptors detect a stimuli, as we've said, but there are specific receptors that you need to learn about. And each receptor responds to a different stimulus, and this stimulus of a receptor leads to the establishment of a generator potential, which can then cause a response. And we'll come to the idea of establishing a generator potential and an action potential shortly. So the 3 receptors that you need to know are the Pacinian corpuscle, rod cells, and cone cells, both of which are in the retina in the eye. So your Pacinian corpuscle, this receptor responds to pressure changes. They are the receptors that you find lots of deep in the skin, mainly in the fingertips and in feet, and it consists of a single sensory neuron, wrapped in layers of lamellae separated by gel. The sensory neuron and obsidian corpuscle has a special channel protein, or has special channel proteins in its plasma membrane. So here's our structure that we can see. And those special channel proteins are stretch-mediated sodium ion channels. And these will open and therefore allow sodium ions to diffuse into the sensory neuron when those channels are stretched and deformed. And when pressure is applied to a fingertip or feet, that is applying pressure to the skin, and these layers of lamellae get pushed and deformed, and that stretches and deforms the shape of the sodium ion channels so that they open and sodium ions diffuse in. So when pressure is applied, it causes these sodium ions to diffuse in, and that will then result in the establishment of a generator potential. More on that very soon. Next, then we look at the rod and cone cells, which are the two photoreceptors, or light receptors in other words, in the human retina. Rod cells process the images in black and white. To create that generator potential, the pigment inside of these rod cells, known as rhodopsin, has to be broken down, and that is done by light energy. They can detect light of very, very low light intensities, as many rod cells connect to one sensory neuron. In this case, we can see it's called a bipolar cell before it gets that sensory neuron. And this is known as retinal convergence because we have multiple rod cells converging or meeting at one bipolar cell. And this means that collectively they are able to trigger that action potential, but because you have got multiple rod cells feeding to one bipolar cell, it means the brain cannot distinguish between the separate sources of where that light energy came from. So that's known as low visual acuity, which means you don't have that sharp, clear, crisp vision. So when it's very, very dark, you can still see, but you only see in black and white, and it's blurry. So next time it's nighttime, have a look and you'll notice you pretty much only see in black and white, and you don't have that clear, crisp vision, meaning low visual acuity, and that is why the retinal convergence. Cone cells process images in colour. There are three types that contain different types of pigment known as iodopsin that you get in the cone cells. You have red iodopsin, green and blue iodopsin, which all absorb different wavelengths of light. Now, iodopsin found in these cone cells is only broken down in high light intensities. So you do need a high light intensity to be able to see in colour vision. You also need to have a high light intensity to see in colour vision because you have one cone cell connecting to one bipolar cell. And therefore, you need to have a high enough stimulus to trigger that action potential. So when it is much brighter light intensities, you can see colour, and you have a high visual acuity because you've got one cone cell connected to one bipolar cell, so the brain can distinguish between separate sources of light. Now, the distribution of the rods and cone cells is uneven in the retina of the eye. And here's the fovea. That is where the highest light intensity hits the retina because of where the light is focused through the lens. So at the fovea, you have most cone cells. We can see here that's where the fovea is. You have lots of cone cells there. Whereas the rod cells, because they still work at lower light intensity, those are mainly located further away from the fovea because they'd still work in that location. Next, then we move on to the control of the heart, and cardiac muscle is myogenic, and that means it contracts on its own accord without any input from the nervous system, but the rate of contraction is controlled by the nervous system and also electricity. So that's what we're going to have a look at. The sinal atrial node, or SAN, is located in the right atrium, and that is known as the natural pacemaker. The AVN, or atrioventricular node, is located near the border of the right and left ventricle. It is still within the atrium, though, just here. You have a bundle of His which runs through the septum, and then the Purkinje fibres branch into the walls of the ventricles. So these two are nodes that will emit the electrical activity. The bundle of His and Purkinje fibres are able to transmit and conduct that electrical activity through the myogenic muscle. So the order that this happens then, the SAN releases a wave of depolarization, which means electricity, across the atria. The AVN will then release another wave of depolarization when that first wave of depolarization reaches it. However, there is a layer of non-conductive tissue between the atria and the ventricles, and this prevents that wave of depolarization travelling straight down through the cardiac muscle in the ventricles. That means instead the wave of electricity has to go through the bundle of His and then the Purkinje fibres. As a result, that means the ventricles actually contract at the apex, which is this very tip here, and then the walls of the ventricles contract after a short delay. And the advantage of this is the atria would have fully emptied the blood before the ventricles contract, and also the ventricles contract from the bottom upwards, so you've got a more efficient pumping of that blood out of the heart. Now, the control of that heart rate is where we see the nervous system taking its role. So the medulla in the brain controls the heart rate via the autonomic nervous system. Autonomic meaning it's automatic, there's no conscious thought behind it. So there's two parts. We have a centre link to the sinoatrial node, so the SAN, to increase the heart rate via the sympathetic nervous system. And then there's also another that decreases heart rate via the parasympathetic nervous system. And that's what we can see here. We've got the medulla, and then we've got these two. We've got the parasympathetic nervous system and the sympathetic nervous system. So the heart rate changes in response to pH and blood pressure. And these stimuli are detected by receptors. Chemoreceptors detect changes in pH, and we have pressure receptors, also known as baroreceptors, detect changes in blood pressure. And both of these receptors are found in the aorta and the carotid artery, which is just after the aorta. So the response to pH, the pH of the blood will decrease, meaning more acidic, during times of high respiratory rate or respiration, because if it's aerobic respiration, there'll be lots of carbon dioxide produced, which will dissolve to make carbonic acid, or if it's anaerobic respiration, you're producing lactic acid. Either way, this excess acid needs to be removed from the blood rapidly, because if it's really acidic, it could denature enzymes and proteins as well, such as hemoglobin. So this is achieved by increasing the heart rate so that carbon dioxide will get to the alveoli, diffuse out via gas exchange more rapidly. So that means there needs to be more impulses via the sympathetic nervous system to the SAN to make that natural pacemaker, the SAN, fire more rapidly. The response to pressure is, if the blood pressure is too high, this can cause damage to the walls of the arteries, and it's important that mechanisms are put in place to bring down that blood pressure to prevent that damage. And that results in more impulses via the parasympathetic nervous system to decrease the SAN firing, so the heart rate decreases. Now, if the blood pressure is too low, there may be insufficient supply of oxygenated blood to respiring cells. So in that case, we'd have more impulses via the sympathetic nervous system to increase the heart rate to bring the blood pressure up. So here is the process. The stimulus in this case we've got is an increase in carbon dioxide, meaning lower pH, and an increase in hydrogen ions. Chemoreceptors in the walls of the aorta and the carotid artery will detect that. Then the coordination is more electrical impulses are sent to the medulla, which is the cardiac centre, and that will then result in more impulses sent via the sympathetic nervous system to the SAN to increase the frequency of electrical impulses. The effector is the cardiac muscle, so the SAN tissue, and the response is an increased heart rate because the SAN will be firing more rapidly. Now, for high blood pressure, the stimulus is increased blood pressure. The receptor would be the pressure receptors in the wall of the aorta and the carotid artery, and these stretch when there's high pressure. Coordination would be more electrical impulses sent to the medulla, and then more impulses sent via the parasympathetic nervous system so that the SAM will fire less frequently. The effector is the cardiac muscle, the SAM tissues, response, reduced heart rate, and that lowers the blood pressure. Next is 3.6.2, nervous coordination. And here we have a motor neuron, which is actually the only neuron that you need to know the structure of. So we've got the cell body of the neuron, and that contains organelles that you'd find in typical animal cells. So the ribosome, nucleus, mitochondria, for example. And proteins and neurotransmitter chemicals are made there. Then we've got the dendrites, which carry action potentials to surrounding cells. The axon is, we can't see it because it's actually surrounded by the Schwann cell, but it's the long, thin, wire-like structure. And it's a conductive long fibre that carries a nervous impulse along the motor neuron. Schwann cells, so these are these Schwann cells here, and they wrap around the axon to form the myelin sheath, which is a lipid and therefore does not allow charged ions to pass through it. There are gaps in these along the axon, and those gaps are known as the nodes of Ranvier. So we then move on to the concept of an action potential and resting potentials. Resting potentials are when a neuron is not conducting an impulse. So there's not been a stimulus detected, it's just your resting standard state. But even when at rest, there is a difference between the electrical charge inside the neuron, specifically we tend to talk about the axon, and the outside of the axon. And that difference in voltage is our resting potential. So there are more positive ions, so sodium ions and potassium ions, outside compared to inside, and therefore the inside of the neuron is comparatively more negative at minus 70 millivolts, and you do need to know that value. So this is quite a common 2 or 3 mark question to explain how that resting potential is established or maintained. So first of all, there is a sodium potassium ion pump, which involves active transport and therefore ATP. So that's what we can see here in the membrane of the axon. So this co-transporter pump, we have 2 potassium ions being pumped in and 3 sodium ions being pumped out by active transport. That creates an electrochemical gradient, so we end up with more potassium ions inside of the cell and more sodium ions outside of the cell. Because we have this electrochemical gradient, those ions can then move down their concentration gradient. But the membrane is more permeable to potassium ions because there are more of these potassium ion channels, and also there's some that are permanently open, whereas the sodium ion channels, there are fewer of them and some aren't always open. So although you have sodium ions diffusing in, you have far more potassium ions diffusing out, and that's why it's more positive outside than it is inside the axon, and you get this minus 70 millivolts. Just point out that is the minus there. That minus has just gone on to a different row. Now, an action potential is when there is a response to a stimulus, and you only get this response to the stimulus if the stimulus is big enough to increase the neuron's voltage beyond a set point, a threshold. So an increase in voltage or depolarization is due to the neuron membrane becoming more permeable to sodium ions, and therefore it becomes more positive. And once an action potential is generated, it will happen initially at this point, and then it moves along the axon from node to node of Ranvier like a Mexican wave. And we'll come back to that concept once we go through the action potential. So here we can see we've got our resting potential at minus 70 millivolts, but then we have a stimulus. And the stimulus provides energy that can cause the sodium voltage-gated ion channels, so those voltage-gated sodium ion channels, to open. And if those open, we then start to get sodium ions diffusing into the axon, which increases the positivity inside of the axon. So we can see that it is becoming more positive. When you have the threshold potential reached of minus 55 millivolts, that then causes even more sodium ion channels, but they're voltage gated sodium ion channels, to open. So we get even more depolarization because more sodium ions diffuse in, up to a maximum of positive 40 millivolts. And at that point, when we get to positive 40 millivolts, that causes the voltage gated sodium ion channels to close, and we still have potassium ion channels open. So we've got potassium ion channels permanently open, so you constantly have positive potassium ions moving out, but now at this point we no longer have positive sodium ions moving in. And that's when repolarization occurs, so it starts to become more negative. And we get even more potassium ion channels opening. So even more positive ions are now moving out and none are moving in. And that's when we get this overshoot, this hyperpolarization going below the resting potential to at least minus 80 millivolts. And this is then when we'd have that resting potential being re-established through active transport with the sodium ion potassium ion co-transporter pump to bring it back to its resting state. So a stimulus has to be large enough to cause the initial voltage gated sodium ion channels to open to reach the threshold. And once that threshold is reached, an action potential will always occur. But if it's a very small stimulus and that isn't enough to open the voltage gated sodium ion channel, then you don't reach the threshold, and it's known as this failed initiation. You don't get an action potential and it goes straight back to resting. Now that is a good thing because we don't want to respond to every single slight change in our environment, so we just overwhelm our senses. We only want to respond to those stimuli that are large enough, that are worthy to respond to, so that it could potentially cause danger and therefore it is worth us responding to it. Now, the refractory period is just labelled here, but you actually have the refractory period happening through all of this point when you've reached that action potential, and we'll come back to the refractory period in a minute. But just showing you this concept of the Mexican wave. So that action potential that we just saw here, this curve, will happen at all of these points along the axon at the nodes of Ranvier. So it's like a Mexican wave because we have this increase and that triggers then an increase here, and that triggers it here, here and here. So each time you reach that threshold, that can then trigger an action potential at the next node of Ranvier. So the all or nothing principle, if the depolarization does not exceed the minus 55 millivolt threshold, an action potential does not occur. So we previously talked about that, and that's the nothing part of the principle. Any stimulus that does trigger depolarization to at least that minus 55 millivolt threshold will always trigger an action potential, and it will always peak at the same maximum. Bigger stimuli does not increase the voltage any further. Instead, it increases the frequency at which the action potentials are generated. So that's the all or nothing principle, and it's important because it makes sure that animals only respond to these large enough stimuli rather than responding to every slight change in our environment. The refractory period, so after the action potential has been generated, the membrane enters this refractory period, which is when it cannot be stimulated to go through another action potential. And it's because the sodium ion channels are recovering and can't be opened. So that should say ions there, sodium ion channels. Now this is important because it ensures that discrete or separate impulses are produced. An action potential cannot be generated immediately afterwards, so it has to be separate, and that makes it easier to process the stimulus. It also ensures that the action potential only travels forwards because it can't go backwards because those sodium ion channels are recovering. So it moves forwards, constantly moving its way around the reflex arc or the nervous pathway. And also it limits the number of impulses, and this is important to prevent overreaction to a stimulus. The factors affecting the speed of conductance are myelination and saltatory conduction, axon diameter, and temperature. Myelination and solitary conduction is linked to the importance of these Schwann cells, which create the myelin sheath. So Schwann cells wrap around the axon to form that myelin sheath, which does not allow charged ions or the impulse to pass through it. So that leaves you with gaps where action potentials can be generated, known as the nodes of Ranvier. And as a result, the action potential jumps from node to node, and that is called saltatory conduction. And this is faster because that means you only have to generate action potentials at these nodes of Ranvier rather than every single position along the axon, and that is why it travels faster. A wider diameter will increase the conductance, and that's because it offers less resistance to the flow of ions inside of the axon, so depolarization spreads more quickly. A higher temperature increases the speed of conduction for two reasons. That conductance will be faster because the ions diffuse faster because they've got more kinetic energy. And also the enzymes involved in respiration will be working faster. So that means there's more ATP for active transport of that sodium ion, potassium ion pump. Synapse is the gaps between the end of the axon of one neuron and the dendrite of another. And that is where the action potential is then transmitted as neurotransmitter that diffuses across the synapse. This here is the whole process of what happens at a synapse. This is worth pausing, screenshot, and you can turn it into a flow diagram, mind map, because this would be a long answer question. So the action potential arrives at the synaptic knob. Depolarization of the synaptic knob causes calcium ion channels to open, and therefore calcium ions diffuse into the synaptic knob. That causes vesicles that contain the neurotransmitter to move towards and fuse with the presynaptic membrane, and that releases the neurotransmitter into the synaptic cleft. That neurotransmitter diffuses down its concentration gradient across the synaptic cleft, and it will bind to receptors on the postsynaptic membrane. When it binds to the receptors on the postsynaptic membrane, that causes sodium ion channels to open. Therefore, sodium ions will diffuse into that postsynaptic membrane of the neuron. And if enough neurotransmitter is bound to cause enough sodium ions to diffuse in to reach that minus 55 millivolt threshold, then it's going to generate that action potential. Lastly, that neurotransmitter needs to be broken down so that it is then released from the receptor to cause the sodium ion channels to close. Because otherwise, even after the stimulus has stopped, that will still be triggering an action potential in the postsynaptic membrane, which we don't want to happen. So that is then going to happen because of enzyme. Enzymes will degrade the neurotransmitter, causing them to be released. Therefore, the sodium ion channels close and it goes back into the resting potential stage. And the neurotransmitter that's been broken down gets recycled and reused. Now, the bits that are in bold and yellow are the parts that I've seen repeatedly in mark schemes that you have to say those terms, and sometimes they're even underlined that you have to use literally that language. So make sure you're really specific with how you word this. A cholinergic synapse is a particular synapse with the neurotransmitter acetylcholine. So it's exactly the process we just went through, except you need to say acetylcholine whenever you would have said neurotransmitter. And you need to know that the enzyme that breaks down the acetylcholine is acetylcholine esterase. And that will break down or hydrolyze the acetylcholine into acetate and choline, which then gets recycled. Summation is what happens at a synapse, and it's the rapid buildup of neurotransmitter in the synapse to help generate an action potential, because we have to have enough neurotransmitter to cause enough sodium ion channels to open for enough sodium ions to diffuse in to trigger and generate that action potential. So the two methods are spatial or temporal summation. Spatial summation is when you have many different presynaptic neurons converging at one postsynaptic neuron. So collectively, these multiple presynaptic neurons release so much neurotransmitter that it should result in the generation of an action potential. Temporal summation is when you just have one presynaptic neuron connecting to one postsynaptic neuron. And in this case, it is the repeated release of neurotransmitter from that one presynaptic neuron over a short period of time that should add up to enough neurotransmitter to cause enough sodium ion channels to open to reach that threshold potential in the postsynaptic neuron. Now, the movement at a synapse is unidirectional. The impulse only travels in one direction. And there are a few reasons why that's the case. Number one is the neurotransmitter is only released by the presynaptic neuron, and therefore you get this concentration gradient. So it can then only diffuse from that location to where there's a lower concentration at the postsynaptic neuron. And secondly, you only have the receptors that the neurotransmitter binds to on the postsynaptic neuron's membrane. You can also get inhibitory synapses, which will prevent an action potential being generated. And these can cause chloride ions to diffuse in instead of or in addition to sodium ion channels. And as a result, because you've got chloride, which is Cl minus, you have negative ions moving in, and that makes that postsynaptic membrane potential even more negative. It could go down to minus 80 millivolts going into hyperpolarization. That makes an action potential highly unlikely because you need even more sodium ions to diffuse in before you'd reach that minus 55 millivolt threshold. A neuromuscular junction is very similar to a synapse, except instead of it being the gap between two synapses, it's actually the end of your nervous pathway where you have a gap between your motor neuron and your effector, which is a muscle. And striated muscle will contract when it receives an impulse from that motor neuron through that neuromuscular junction, and it works in a very similar way to synapses. So first of all, an action potential reaches that presynaptic membrane of the motor neuron, causing calcium ion channels to open, calcium ions diffuse in. That causes the vesicles that contain the neurotransmitter to move towards and fuse with the presynaptic membrane, releasing the neurotransmitter, which is acetylcholine, that diffuses across this gap, and then it will bind to receptors on the postsynaptic membrane. But the postsynaptic membrane here is the sarcolemma, which is a membrane of the muscle fibers. The binding causes sodium ion channels to open in that sarcolemma and sodium ions diffuse in. So it's pretty much identical, except we've got not a postsynaptic neuron, we've got muscle fiber, so that receptors are on the sarcolemma, and it's always going to be acetylcholine as the neurotransmitter. So that depolarizes the membrane of the muscle fiber and creates an action potential in that muscle that moves along these T tubules towards the center of the muscle fiber. And those action potentials prompt the opening of voltage-gated calcium ion channels in the membranes of the sarcoplasmic reticulum, situated near these T tubules. And as a result, calcium ions diffuse out of the sarcoplasmic reticulum around the myofibrils. And that is what causes muscle contraction via the sliding filament. Now this flows into what we were just talking about, muscle contraction, 3.6.3, skeletal muscles and looking at how they can contract. So we talked about the myofibril. Here are the structures that you need to be aware of. Again, this could be a good one to screenshot, turn into flashcards for each of these structures. The myofibril, these are long cylindrical structures found inside muscle fibers, made up of repeating sarcomere units, which we're going to have a look at. Because the sarcomere is the functional unit, it's defined as a region between two Z lines, and you have actin and myosin where you can have your sliding filament theory occurring. The sarcolemma, that's the plasma membrane of a muscle fiber, It conducts electrical impulses, the action potentials, and it triggers the contraction to help maintain ion balance within the cell as well. Sarcoplasmic reticulum, that's a specialised form of smooth endoplasmic reticulum that surrounds myofibrils, and it stores and releases calcium ions, which are essential for triggering muscle contraction. The sarcoplasm, that is the cytoplasm of a muscle fibre. So it contains the organelles, enzymes, myoglobin needed for energy release and muscle function, and it also has glycogen stores for ATP synthesis. So looking at the sarcomere in a bit more detail, this is what we said on the previous slide. So this is one long cylinder, which is the myofibril, made up of these repeating units called sarcomeres, which are identified by two Z lines. There are other structures within the sarcomere that you need to be able to name. So here are the Z lines, which indicate the start and end of one sarcomere. The A band is the myosin. So that is the distance of myosin, which is shown in blue here. An I band is the distance where you have actin not overlapped at any point with the myosin. So just red lines here, which are our actin protein. We have an M line, which is the center of where the myosin is attached. We have an H zone, which is where you just have myosin without any actin overlapping it. So those are your key structures, all to do with basically whether you have actin by itself, myosin by itself, and where it's overlapping. And the kinds of questions you might get is, what happens to the size of those sections when a muscle contracts? And when a muscle contracts, the actin slides inwards over the myosin, which we can see here. So as a result, the H zone decreases in length because we now have less myosin by itself. The I band will also decrease in length because we have less actin by itself because it's slid over. The A band never changes because that is the length of the myosin. Myosin doesn't change in length. The M line will always stay in the same position as well, but the Z lines move closer together. So that's something you need to know as well, those different bands and what happens to them when a muscle contracts. The sliding filament theory explains why you have the actin sliding over the myosin. So when that action potential reaches a muscle fibre, like we saw in the neuromuscular junction, it triggers the release of calcium ions from the sarcoplasmic reticulum into the sarcoplasm. And those calcium ions cause tropomyosin, which is a protein that is normally blocking the binding sites on actin, to move, and it exposes the binding sites for myosin on actin. Now, because of that, we get this cross bridge formation. So with ADP and PI still attached to our myosin head, which is a structure branching off myosin, that can now bind to the myosin binding sites on actin. And when that happens, it creates what's known as an actino-myosin cross bridge. The power stroke, which is when the actin filament is moved, once that cross bridge is formed, the myosin head pivots, so it bends here, and that pulls the actin filament inwards, so it slides it in. And that movement is known as the power stroke, and when that happens, it causes the release of ADP and PI. It also generates tension, and that is causing the actin filament to slide. So we then get this ATP molecule binding, and that new ATP molecule, when it binds to the myosin head, it causes it to detach from the actin filament. And this ensures that the myosin head doesn't remain permanently attached to the actin. That ATP then gets hydrolyzed, and it resets the myosin head. So within the sarcoplasm, there is the enzyme ATPase, and that is activated by calcium ions. That will hydrolyze the ATP back into ADP and PI, and the energy released from that ATP hydrolysis repositions the myosin head back to its original state, ready to form a new cross bridge, and therefore go through that process again to bring the actin even closer for a full muscle contraction. So as long as calcium ion levels remain high, the cycle repeats, continuously pulling the actin filaments towards the centre of the sarcomere, shortening the muscle fibre and causing contraction. When the nervous system stimulation stops, calcium ions are then actively transported back into the sarcoplasmic reticulum, and that allows the tropomyosin to move back to block the myosin binding sites on actin to prevent any further contraction, and the muscle will relax. So some common questions. What's the role of ATP and phosphocreatine? Here are your standard answers if this came up. So the ATP is hydrolyzed and the energy will then break the actinomyosin bridges, or it's to move or bend the myosin head back to its original state, and for the active transport of those calcium ions. So if ATP levels are low, muscle contraction can't happen effectively. And that is where phosphocreatine plays a role, because this acts as a rapid source of ATP in muscle cells by donating a phosphate group to ADP to be able to make ATP, so that you do have ATP during short bursts of intense activity. You could also be asked what's the role of glycogen granules in skeletal muscles. So it's a store of glucose that can be hydrolyzed into glucose, and that glucose can be used as a respiratory substrate to provide the ATP needed. The role of calcium ions then, so when an action potential reaches the muscle fibre, calcium ions are released from the sarcoplasmic reticulum and diffuse into the myofibrils. Those calcium ions cause tropomyosin to move and expose the binding sites on the actin filament. And with these sites now accessible, the myosin heads attach to actin, forming the actinomyosin cross bridge. The last bit of this topic is knowing the difference between slow twitch muscle fibres and fast twitch muscle fibres. So knowing the difference between the structure, location, general properties, the type of respiration that happens, ATP production and energy storage. So I recommend screenshot this page, or if you've downloaded my free booklet, fill it in so you've got this for your revision. So in terms of structures, slow twitch, large store of myoglobin, rich blood supply with many mitochondria, compared to fast twitch, thicker with more myosin filaments, large store of glycogen and a store of phosphocreatine. Location, you get a lot of slow twitch in calf muscles, whereas fast twitch might be places like biceps. General properties, slow twitch contract slower, and they can sustain aerobic respiration for longer periods. So these muscles are used in endurance activities like marathons. Fast twitch contract faster and provide short bursts of powerful contraction for things like sprinting or weightlifting. Slow twitch will mainly be aerobic, fast twitch is anaerobic, and in terms of ATP production, the slow twitch produce ATP slowly, but they'll generate more, and that's because it's aerobic. And because fast twitch are anaerobic, they produce ATP quickly, but less. Lastly, in terms of the glycogen store, they both have stores of glycogen, but slow twitch will have a lower store of glycogen, and you'll have a large store in the fast twitch. Next then is 3.6.4, homeostasis. Homeostasis involves physiological control systems that maintain the internal environment within restricted limits. And it's important in maintaining things like a stable body temperature, stable blood pH for enzyme activity. Negative feedback is involved, and this restores systems to their original level. And the possession of having separate mechanisms, so one that can bring levels back up, one that can bring it back down, give better control to get it back to the original state that it should be at. So for body temperature, if it's too low, there'd be insufficient kinetic energy for enzyme-controlled reactions. If it was too high, enzymes would denature. Either way, metabolic reactions would stop or slow and cells would die. If you have too high or too low a blood pH, it will result in enzymes denaturing, fewer enzyme substrate complexes, and again, that are going to affect metabolism. Blood glucose control. So glucose is needed for respiration. So a lack of glucose in the blood could result in cell death. And if blood glucose levels are too high, then this lowers the water potential of the blood, and water then leaves the surrounding cells by osmosis, and that prevents the normal function of those cells because now they're low on water. And also it increases the blood pressure because there's more liquid in the blood. The water potential of the blood then, if it's too high, water moves into cells by osmosis and that could cause them to burst. And if the water potential is too negative, the water leaves the surrounding cells into the blood and that can cause the cells to crenate. So you need to know in detail blood glucose concentration. And that is going to increase after you've eaten or drunk something containing carbohydrates, or it will decrease if you haven't eaten something containing carbohydrates or following exercise, because respiration rates would be higher using up your carbohydrates. Here are the different things involved. So the pancreas, that is the organ that detects changes in blood glucose levels. And specifically, you have the islets of Langerhans cells that are going to detect and release insulin and glucagon to bring blood glucose levels back to normal. Insulin is a protein hormone released when blood glucose levels are too high, so it causes a decrease in blood glucose levels. Glucagon is also a protein hormone and it's released when blood glucose levels are too low, and that will cause an increase in blood glucose levels. Now, adrenaline, this is released by the adrenal glands when your body anticipates danger. So it's not to do with homeostasis, but it has a very similar effect to glucagon because it causes glycogen to be hydrolyzed, so you increase the blood glucose levels. So some key terms that you need to know for this topic are glycogenesis, glycogenolysis, gluconeogenesis. So learn those terms, get them on a flashcard. Wherever you see glyco, that means it's about glycogen. So these top two are about glycogen. Where you see gluco, that means it's about glucose. Genesis means to make, lysis means to break. So glycogenesis means making glycogen. So it's the process when excess glucose is converted into glycogen. And that happens in the liver quite a lot and muscle cells. Glycogenolysis, so glycogen lysis, is the hydrolysis of glycogen into glucose. Gluconeogenesis. Gluco means glucose, genesis means to make, neo means new. So it's making glucose from something new. And by that we mean a non-carbohydrate source. So for example, from amino acids, or it could be from glycerol from lipids. So your normal blood glucose levels might increase if you've just eaten a carbohydrate-rich meal. That is detected by the beta cells in the islets of Langerhans in the pancreas. The beta cells will then release insulin. That causes liver cells to become more permeable to glucose, so more glucose is taken from the blood into the liver cells, and then that glucose is converted into insoluble stores of glycogen. That means that we've removed glucose from the blood and brought our blood glucose levels back to normal. Now, maybe you haven't eaten anything containing carbohydrates for a while or you've done high levels of exercise, so lots of respiration using up the blood glucose levels, so they decrease. A decrease is detected by the alpha cells in the islets of Langerhans of the pancreas. In response, the alpha cells release glucagon. That triggers the second messenger model, which activates enzymes to hydrolyze glycogen in the liver back into glucose, which is then released into the blood to bring it back to normal. But we're going to go through that in a bit more detail. So here's our action of insulin. We said that's the hormone released by the beta cells in the islets of Langerhans when blood glucose levels are too high. And insulin will then be transported in the blood. When it gets to the liver cells, it will bind to complementary shaped receptors on the cell surface membrane of those liver cells, also muscle cells. When it binds, it increases the permeability of that membrane to glucose by stimulating the insertion of more glucose channel proteins, GLUT4, into the membrane. And that allows more glucose to move from the blood into the liver cells by facilitated diffusion. Now, once that happens, the glucose has to be converted to glycogen because that is insoluble and therefore it's not going to lower the water potential of the cell like glucose would, and therefore it won't affect osmosis. So these actions help to reduce the blood glucose levels back to normal via negative feedback. And this is just showing us here when the insulin binds to complementary shaped insulin receptors embedded within the membrane of the target cell, the liver cell. That causes a cascade of chemical reactions, which then causes vesicles, which contain the glucose channel protein, to move up and fuse with the cell surface membrane, and it embeds more of those channel proteins, so more glucose can then move from the blood into the cell. The action of glucagon then, the alpha cells in the islets of Langerhans detect the blood glucose level is too low and secrete glucagon, which will travel around in the blood, and when it gets to the complementary shaped receptors on the cell surface membrane of liver cells, it activates a second messenger model, which is described here. So it causes a protein to be activated into adenylate cyclase. That is converting ATP into cyclic or cyclic AMP. Cyclic AMP activates another enzyme, protein kinase, and that's the one that hydrolyzes glycogen into glucose to release it back into the blood. Now, if your glycogen stores are used up, then you can also have the activation of enzymes that can convert glycerol and amino acids into glucose to be used as a respiratory substrate instead. So here's our second messenger model. The first messenger is glucagon. That hormone binds and it activates enzyme number 1, which is adenylate cyclase. Now, adenylate cyclase is going to convert ATP into cyclic AMP, which is our second messenger. And that will then activate the protein kinase, which can hydrolyze glycogen into glucose. Now, adrenaline also goes through that same second messenger model, so you get that same process happening. Diabetes, you have type 1 and type 2 diabetes. Type 1 is when you're unable to produce insulin. Usually starts in childhood, and it could be a result of an autoimmune disease where the beta cells are attacked. So the treatment would be injections or some way of getting insulin into the blood. Type 2 diabetes is when the receptors on the target cells lose their responsiveness to insulin. So insulin is still made, it's just the receptors don't respond as they should. And this usually develops in adults because of obesity and poor diet. It can be controlled by regulating the intake of carbohydrates, increasing exercise to use up that glucose for respiration, and sometimes insulin injections are still needed as well. Osmoregulation. So this is now looking at the regulation of the water potential of the blood. And blood with too low a water potential is described as hypertonic. And that might be if you've been sweating lots, not drunk enough, or there's lots of ions in your diet. Hypotonic is if the blood has too high a water potential, and maybe you've drunk too much water or you don't have enough salt in your diet. The corrective mechanisms would be, if you've got too low a water potential, then more water is reabsorbed by osmosis into the blood from the tubules of the nephrons in the kidneys. So that would mean you make urine, which is more concentrated and lower volumes. If you've got too much water in your blood, so too high a water potential, then less water is going to be reabsorbed by osmosis back into the blood from the nephrons. So you end up with larger volumes of dilute urine. So let's have a look at this nephron then. Osmoregulation occurs within the nephrons in the kidneys, and nephrons are these long tubules surrounded by capillaries, and there are around 1 million per kidney. The blood gets filtered here to remove waste and selectively reabsorb useful substances back into the blood. So the key stages, which we will go through each in more detail, we have ultrafiltration first, and that occurs due to high hydrostatic pressure. That will force out from the blood water and small molecules like glucose. And that gets forced from the glomerulus capillaries into this renal capsule. Then in the proximal convoluted tubule, we have selective reabsorption occurring. And that is where we're going to have all the glucose and some water being reabsorbed. Then we've got the loop of Henle, and this maintains a sodium ion gradient, so water can be reabsorbed into the blood. And water then will continue to move out of the distal convoluted tubule and collecting duct by osmosis, and the collecting duct carries the remaining liquid, which is the urine, to the ureter. So the structure of the nephron then, the formation of the glomerular filtrate, ultrafiltration, that occurs here in the renal or Bowman's capsule. And there is a high hydrostatic pressure in these capillaries, the glomerulus, and as we said, that forces out water, glucose, ions and urea from the blood into that capsule, large molecules like proteins and red blood cells remain in the blood. So here we have what causes that high hydrostatic pressure first of all. As the blood enters the glomerulus via the afferent arteriole, which is a wider blood vessel, the difference in that size creates a high pressure inside the glomerular capillaries, and it's the high hydrostatic pressure that forces out all of those small solutes and the water. Now you do have 3 filtration barriers. We first of all have the capillary endothelium. So the glomerular capillaries have fenestrations, which are these tiny gaps between the endothelial cells. And that will allow things like plasma and dissolved substances to pass through, but it blocks the red blood cells. So by plasma, I mean it'd be like the water, urea, and glucose amino acids, but blood cells can't fit through there. The next layer is the basement membrane. And this is a gel-like protein-rich layer between the capillary and the Bowman's capsule. It acts as the main filtration barrier and it prevents the passage of the large proteins. So small molecules can pass, but large proteins cannot. And then finally, we have podocytes. And podocytes have foot-like extensions that wrap around the capillaries that create these gaps, these filtration slits, to further ensure only small molecules can enter. So that's how we have the filtration. Then we have the selective reabsorption. So all glucose is reabsorbed by active and co-transport, and most water is reabsorbed by osmosis at this stage. The proximal convoluted tubule, which it says PCT here, do not abbreviate that in the exam. It is not an accepted abbreviation. The proximal convoluted tubule has cells have many microvilli and mitochondria to maximize this active transport and co-transport. So those cells lining the proximal convoluted tubule actively transport sodium ions out of the epithelial cells into the blood via sodium-potassium pump. That creates a lower concentration of sodium ions in the proximal convoluted tubule compared to the lumen. And therefore sodium ions diffuse into the proximal convoluted tubule down their concentration gradient, but that has to be via a co-transporter protein which carries glucose with it as well. So that's how we get the glucose inside of the proximal convoluted tubule, epithelial cells. And then once inside there, the glucose moves by facilitated diffusion into the blood, and that is how all of the glucose re-enters the bloodstream. So adaptations include these cells have microvilli, so these highly folded cell surface membranes to increase the surface area for that transport. They also contain many mitochondria so that lots of ATP can be produced for this active transport. And then there'll also be lots of carrier proteins and co-transporter proteins embedded in the cell surface membranes for all of that transport. And then water is reabsorbed by osmosis because following the absorption of the solutes, that creates a water potential gradient, so then water can move in by osmosis as well. Then we get to the loop of Henle where we have the maintaining the sodium ion gradient. And in the descending limb, water leaves by osmosis, but sodium ions cannot. So the descending one is the one going down. The ascending limb is the one going up. Sodium ions are actively transported out, but water cannot leave. And this creates a water potential gradient in the medulla, so this surrounding area outside of the nephron, which allows even more water to be reabsorbed by osmosis at the distal convoluted tubule and the collecting duct. And water reabsorption is regulated by the hormone ADH, which we're going to come on to shortly. So here's a bit about the loop of Henle in a bit more detail, because we said that descending limb, the walls are thin and permeable to water, but impermeable to ions. So as the filtrate moves down the descending limb, water will move out by osmosis into the surrounding medulla due to that lower water potential, and the concentration of the filtrate will increase. As it moves up, the filtrate moves up the ascending limb. The walls are thicker and they're also impermeable because they don't have aquaporins within them, so water cannot move out by osmosis. But sodium ions can be actively transported out, And that then means we get a really negative water potential gradient. Now at the base of the ascending limb, where the filtrate is very concentrated, some sodium ions will also move out by facilitated diffusion. But this whole process creates a high concentration of sodium ions in the medulla, lowering the water potential, which is essential for water reabsorption in the distal convoluted tubule and collecting duct. So how the hypothalamus and the posterior pituitary gland are involved. The hypothalamus is going to detect changes in the water potential. Specifically, we have osmoreceptor cells, which are the receptors that detect the changes in water potential. So if the water potential of the blood is too low, water is going to leave the osmoreceptors that are in the hypothalamus, causing those cells to shrivel, and that stimulates the hypothalamus to produce more of the hormone ADH, antidiuretic hormone. And if the water potential of the blood is too high, water enters the osmoreceptor cells by osmosis, and that stimulates the hypothalamus to produce less ADH. Now, the hypothalamus is where ADH is produced, but ADH then moves to the posterior pituitary, and that is where it is released from into the blood. You do need to know that distinguishment. It's come up as an exam question before. And once that ADH is released in the blood, it then travels in the blood to its target organ, which is the kidney. So ADH, antidiuretic hormone, this is going to cause an increase in the permeability of the walls of the collecting duct and the convoluted tubule. And as a result, more water is going to move from the nephron by osmosis back into the blood. And that results in urine that is more concentrated and in smaller volumes. So there are receptors on the cell membranes of the distal convoluted tubule and the collecting duct to which ADH bind. When ADH reaches the kidney, it binds to those receptors. And when bound to the receptors, it activates a phosphorylase enzyme in the cells. The phosphorylase causes the vesicles containing aquaporins to move and fuse with the cell membrane, so you get more of these aquaporins, which are protein channels that water can move through. Now, this is a little bit extra information. You could include it in an essay. You could still get marks sometimes in the mark scheme for it, but it's not essential that you need to know that process. You do need to know that ADH binds to the receptors, increases the permeability of the cell membrane to water. Aquaporins is often a mark as well. So therefore, ADH causes an increase in the permeability of the walls of the collecting ducts and the convoluted tubule, which means more water will move into those cells and then into the blood by osmosis. And therefore, urine is more concentrated and you return more water to the blood. So in summary then, if the water potential of the blood increases, meaning too much water, so you might have drunk a lot of water, that's detected by the osmoreceptors in the hypothalamus. Hypothalamus produces less ADH. The distal convoluted tubule, which again, that is not an accepted an abbreviation. Do not abbreviate it like that in the exam. You have to write it out in full. ADH you can abbreviate though. So the distal convoluted tubule and the collecting duct walls become less permeable to water. So that means less water is reabsorbed in the blood and more remains in the filtrate in that collecting duct and goes on to create larger volumes of more dilute urine, bringing your water potential of the blood back to normal. Now, maybe you've sweat a lot and you've lost a lot of water and your water potential of the blood decreases. That is going to be detected by the osmoreceptors in the hypothalamus. The hypothalamus will produce more ADH, which is released into the blood by the posterior pituitary. The distal convoluted tubule and the collecting duct walls become more permeable to water, so more water is reabsorbed into the blood by osmosis and less is lost in the urine. So you get more concentrated urine, and that means you get your normal water potential of the blood. So that takes us to the end of topic 6. If you found this helpful and you want even more help with the mark scheme points, then try out my flashcards. If you want full details, plus mark scheme points highlighted, key terms, essay link, recall question and examiner tips, then scan here to try out my A-level notes. Both also linked in the description. But that is it for today's video. Hope you found it helpful and I'll see you in a video very soon.